Yeah.
We are about to begin our panel discussion. I once again request all the audience to get back to indoor.
Uh, oh. It's okay. Yeah. Catch.
Good evening to one and all present here. Science is a systemic way of exploring the world and explaining it to satisfaction of the evidence. So goes the quote from Stephen Hawking. famous English theoretical physicist. Sharing knowledge is a way to attain mortality.
Respected dignitaries present, faculties, and my dear friends, we welcome you all to the panel discussion session of BioZion, the Biotech Capstone Science Society Interface. Here, our experts have gathered to express their opinions, challenge each other, explore, and provide valuable insights on biotechnology at 2047.
Now let us introduce the panelists for today's session. First we have Professor Dr. Habil Thomas Braun, Director, Max Planck Institute for Heart and Lung Research, Bad Nauheim, Germany. He has a PhD in medicine and received German habilitation in biochemistry. He served as a professor at the University Halle-Wittenberg and director of the Institute of Psychological Chemistry. Dr. Braun is also a professor at medicine at Justus Liebig University, Gießen. His research focuses on the development and regeneration of the heart and skeletal muscle, as well as the molecular mechanisms of congenital heart disease. He has published more than 300 papers and received several awards for his work. Sir, we are honored to be in your presence.
Next, we have Dr. Sanjay Behari, Director, Sri Chitra Tirunal Institute for Medical Sciences and Technology, former Professor and Head of Neurosurgery, Sanjay Gandhi Postgraduate Institute of Medical Sciences, and also a Fellow of Royal College of Surgeons, Edinburgh, American Association of Neurological Surgeons, and Academy of Medical Sciences, India. His expertise include skull base and neuro-oncology, complex spine and craniovertebral junction, and vascular neurosurgery. He did his MBBS with honors and has received Best Student Award from SMS Medical College, Jaipur. He completed his MS in General Surgery with 2 gold medals in Chief of Surgery and Best Scientific Paper. He obtained his MCH and DNB in Neurosurgery from SGPGI, Lucknow. He was also a Sugita International Fellow at Nagoya University, Japan. We are glad to have you here, sir.
Professor G.M. Nair is a prominent figure in the field of academia and biotechnology research. He is currently serving as the director of the Central Laboratory for Instrumentation and Facilitation, University of Kerala. In this role, he has been instrumental in fostering an environment of excellence in research and education, nurturing young minds and promoting interdisciplinary collaborations. He also held a distinguished role as an advisor to the Kerala Biotechnology Commission, playing a pivotal role in shaping biotechnological advancements in the state. Additionally, he served as a former director of the Tropical Botanic Garden and Research Institute, TBGRI. We welcome you, sir.
Moving forward, we have Dr. Achut Shankar, Professor of Computer Science in University of Kerala, who heads the Department of Bioinformatics. He is a teacher, researcher, and a science communicator with more than 3 decades of academic experience in India and abroad. He was a director of CDET, Government of Kerala during 2001 and 2004. He had his education from College of Engineering, Trivandrum, IIT Bombay, University of Cambridge, and University of Kerala. He holds 2 PhDs, one in engineering and one in music. Under his leadership, the whole genome sequence of 2 plants, Trichopus zeylanicus and Clitoria terrentia, have been sequenced and published. He has successfully guided 30 PhD students. He is a recipient of the first National Teachers Award by the Indian National Science Academy, New Delhi. He is also a recipient of Vaikam Maulavi Foundation's Award for Social Service. We welcome you, sir.
Next, we have Dr. Rajiv K. Sukumaran, Senior Principal Scientist and Head, Microbial Processes and Technology Division, CSIR, National Institute for Interdisciplinary Science and Technology, Trivandrum. He is currently heading the Biofuels and Biorefinery section of Microbial Processes and Technology Division at CSIR, NIST. He is a member of the Kerala State Biotech Commission and an expert for monitoring and controlling the activities of State Center Resource Institute for Partnership in Technology. He did his master's and PhD in biotechnology at the Cochin University of Science and Technology. He had postdoctoral trainings in molecular immunology at Mount Sinai School of Medicine, New York, and in stem cell biology at National University Hospital, Singapore. Since then, he has been working on biomass conversion to biofuels, especially on enzymes for biomass hydrolysis. His work on cellulases had led to the technology transfer on the production of this enzyme and development of highly efficient beta-glucosidase enzyme for biomass hydrolysis. We are glad to have you here, sir.
Next, I introduce Dr. Sagar Pandit, Assistant Professor, Indian Institute of Science, Education and Research, Pune, German Academic Exchange Services and Research Ambassador as well. He worked as a group leader, Tritrophic Interactions Group, Department of Biochemistry, and also a group leader, Lepidopteran Reverse Genetics, Department of Molecular Ecology, Max Planck Institute, Germany. He has over 12 publications in various journals and 2 patents. He was honored with DAD, German Academic Exchange Service Research Ambassador in 2022, and Max Planck India Partner Group Headship in 2016. DPT Ramalingaswamy Re-Entry Fellowship in 2015-16, and he was elected as a Life Fellow of the Entomological Society of India in 2021, and Life Fellow of the Royal Entomological Society, UK. He is mentoring for agri-tech and biotech sectors for the KBC NMU Center for Innovation, Incubation and Linkages, the Incubation Center of Kavayatri Bahinabai Chowdhury North Maharashtra University, Jalagan.
Up next, we have Dr. K.V. Soni, Professor and Head, Department of Molecular Biology and Biotechnology, College of Agriculture, Velaini. She pursued her master's in biotechnology from Cochin University of Science and Technology and received the K.S. Krishnan Fellowship from the Department of Atomic Energy, BARC. She received her PhD from Amala Cancer Research Center and postdoctoral fellowship at the Indian Institute of Science, Bangalore. She worked as a junior scientist at the Rubber Research Institute of India. After joining Kerala Agricultural University in 1997, she has been greatly involved in teaching, research, and extension activities, and also has implemented different projects in biotechnology. She has also participated in the space biology program of VSSC. She has guided 3 PhD students and 22 MSc students and is currently guiding 2 PhD students. One of the PhD programs was selected by the Kerala Science Congress for the Young Scientist Award. She has a total of 35 research articles published. Ma'am, we welcome you to this session.
Last but not the least, the moderator of our session, Dr. Renu John, Fellow of the Royal Society of Biology. He is currently working as Professor and Head, Department of Biomedical Engineering, IIT Hyderabad. He is also Founder Director and Head, Center for Healthcare Entrepreneurship and Chair at the Center for Interdisciplinary Programs in IIT Hyderabad. He has bachelor's and master's degree in physics and also PhD. He is proficient in optics, biomedical imaging specializations, biomedical imaging and microscopy, 3D imaging, biosensors, and medical devices. He is a postdoctoral research fellow, Beckman Institute for Advanced Science and Technology, University of Illinois, Urbana-Champaign, Illinois, USA, and also postdoctoral research fellow, Duke University, North Carolina, USA. He has an experience of over 18 years in clinical and preclinical imaging studies. He has more than 35 publications overall in various reputed journals. He is a registered fellow at Royal Society of Biology, member at Optical Society of America, and member secretary of the Indian Laser Association. We welcome you, sir. Now we are handing over the session to our moderator, Dr. Renu John.
So, a very warm welcome to all of you panelists for today's evening session. from me. So it's a pleasant evening for all of us to be here in the city of Trivandrum, the city of the Lord Sri Padmanabha and Sri Padmanabha Dasan. And this is also very important that is one of the places from where the first indigenous heart wall was made. Again, we have Dr. Sanjay ji here representing Sri Chitra and as representing the set of pioneering works and innovations from Sri Chitra. I'm also extremely honored to be here on this campus at IIT. In fact, this is probably one of the best agricultural programs in the country. The students from the different campuses, Trivandrum, Thrissur, and Pilikod, and Wayanad also, right? Yeah. So have been contributing to the agriculture scenario of the country and also the advanced biotechnology research. If you look at some of your alumni, not probably all of your alumni, have been representing various national programs, have been scientists at ICAR, and to the grassroots contributing to the KVKs, the Krishi Vignan Kendras, the Krishi Bhavans, and all those where I have never seen anywhere in the country so passionate agricultural officers than in Kerala, who would be talking much more about research in agriculture. So I think this is one of the places where these agricultural scientists have been produced. So you have to be proud of your institute. So when we talk about the biotechnology as such, so I would see that this is one of the areas where everybody have been looking at, where do we look at the next generation of research going to be? What are how the developments in biotechnology are going to affect our social, economic, and scientific outcomes? So the first question to the panel members today would be like, this is a world where biotechnology is increasingly playing a central role in our lives. What are the advances and potential challenges that biotechnology community faces? Or we can say, how are the contributions or advances in biotechnology creating an impact in our in each of our respective areas? So we would start our discussions maybe from Professor Pandit. Yeah. So you can please start looking at how biotechnology has created an impact in your respective areas.
Sure. Thank you first of all for inviting me for this panel discussion and for this entire event. It's a wonderful place to be in, Trivandrum and Kerala especially. So I'm very happy to be here, as I'm always. Thank you for that first question to me and actually giving a good introduction to what I should say, because I come from the plant sciences background, especially agriculture, biotechnology background. And I don't think there is any other field which has experienced the biotechnological push and the biotechnology need as much as the agriculture has as compared to the others. I think what we have seen in recent past, probably if you have heard about BT cotton and BT brinjal, all these things, these are all GMOs which actually masked the other disciplines of biotechnology. So biotechnology is something beyond creating the genetically modified organisms as long as it comes to the plants and our crops. By saying that, we can't take away the credit of creating these interesting organisms. BT cotton revolutionized the entire cotton agriculture. BT brinjal, as we were talking just before this, has faced some socio-ethical and socio-political hurdles, because cotton we wear, but brinjal we eat. And so we are not really ready to accept a GMO for our intake. So basically, socio-ethical and socio-political acceptance is, I would say, the major challenge to the biotechnology in the current era. Other challenges are actually very contradictory, I would say. You know, on one side, the biotechnology has the word bio in it. And whenever it is bio, the process has to be slow, much slower than the chemical and physics related processes. So everyone, including the actual consumer, need to be really patient until the biotechnology delivers. And the contradiction is on the other side, where the scientists are working on developing something, their patience is being tested by the very high level rules and regulations of the governments, and there are a lot of biosafety and bioethical issues that they have to deal with. Although they see the products in their site, these rules and regulations take a pretty long time for them to come to the product stage. Basically, this entire procedure is quite time-taking. And if you ask me, this amount of time that is being taken is the major challenge right now that we have. I would say, apart from this, all other problems, we are pretty ready to take up. Okay. Yeah. So the other problems are pretty easy to deal with. I hope I will stand still here and talk. Okay. So that's all I think I would say regarding the agricultural part.
Yes, Dr. Thomas Braun.
Yeah, just speaking about things being slow until it reaches the consumer, you're absolutely right. I mean, there are a lot of regulations that, for good reasons, have been introduced. I always think of first Nobel Prize in medicine. Yeah, I mean, such inventions made at that time, well, the people would have landed in jail by now. I mean, they also took an incredible risk at that time, and now society isn't really prepared to take risks. So there are a lot of regulations, again, for good reasons. On the other hand, if you just look at COVID recently, how fast vaccine development was, also using a completely new technology, I found that really amazing. Yeah? So when there is the need and the pressure, things can definitely move faster. But you also mentioned acceptance by society, and in Europe, you know, I mean, when it comes to genetically engineered organisms, the whole society is highly skeptical. Yeah, but it's also, in my view, something that's due to simple lack of knowledge. They have this idea of monsters being created by mad scientists in the lab, but they simply lack basic understanding. And I think that's also a responsibility of all of us, just to make the public in general a little bit more aware. Actually, the European Parliament is also discussing at the moment whether these tiny modifications, mutagenesis screens introduced by CRISPR, that this is exempted, and that resulting organisms are not dubbed genetically engineered anymore if you don't leave a trace behind, yeah, like a selection marker or something. I mean, I think it's slowly moving, but I think all the challenges that the world's growing population faces, I mean, this cannot be handled without also appropriate and relatively fast, with all the safety measures, developments. When it comes to medicine, of course, it takes, I mean, if you look at drugs, after first discovery have been made, how long it usually takes until it reaches the patients. I mean, it's usually 20 years or so, yeah. So, and I think, I mean, one should also do a little bit to accelerate that. But on the other hand, I also see there are clear progress made. I mean, just talking about biotechnology and tissue engineering, I mean, there are the first clinical trials ongoing with artificially engineered heart tissue, yeah, for transplantation into and on engineered tissue grafts there on diseased hearts. So things are moving forward, and I think we should also be aware that we are in a very privileged situation. I mean, we have now technologies at hand, from imaging to bioinformatics. I mean, we were completely unimaginable a few years ago. I mean, when I started to do molecular biology, I started off by isolating my enzymes because they were commercially not available. Yeah? So isolated reverse transcriptase restriction enzymes, very slow. Now you can buy them off the shelf, and you have techniques, again, imaging, bioinformatics, advanced genetic engineering methods. And I think we are in the unique position to take advantage of these privileges. And I mean, I also mentioned that I think one should also give freedom, particularly also to young researchers, if they come up with crazy ideas. I mean, of course, at a certain time point, it's absolutely necessary also to protect your intellectual property, to think about also commercial exploitation. But the starting point should always be a cool idea. Boldly go where no one has gone before. Just look for things that nobody thought about. And I think this is then essentially creating really innovation and brings and moves things forward. And I'm pretty sure that all of you here in the audience, all the young faces, if they commit and are motivated, smart, that they will greatly contribute to such progress. And I think this will accelerate even further in the future.
Thank you. Dr. Sanjay Behari ji.
So thank you. It's a privilege and honor for me to be here. In medicine, the potential is immense. First of all, let's start with nutrition. So agriculture and nutrition. So what happens is that if there is a person with a kidney disease, then you have to avoid proteins so that proteins are not excreted. And therefore your diet has to be changed in such a way that proteins have to be eliminated. On the other hand, if you have diabetes, then you have to avoid sugar in your diet and increase the amount of proteins and fibers in the diet. So based on your disease, your nutrition has to change, and we need a lot of supplements. There's another thing which most people don't know, that if you are in the intensive care unit, then the caloric requirement increases almost 5-fold. And there is a negative energy balance. That's why when you have a prolonged illness, you become thin. Because what happens is that your muscle mass is being broken down to give you energy. So therefore, dietetics and nutritional dietetics has a huge role to play in medicine. The second very important area is, of course, stem cells. And the area is developing. Say, for example, somebody has a spinal cord injury, then there is the central nervous system does not regenerate itself. And therefore, you need to give some supplement cells to home into that site and actually create connections with the normal tissue there. The problem is, it is possible to do that, but the only problem is that their functional ability and their ability to interact with other neurons has still not been established. So that is one important area which needs to be focused upon. The third thing is, for example, there are some rare diseases like for Duchenne muscular dystrophy, or you have motor neuron disease, or Parkinsonism. These are continuously progressive degenerative diseases, and there is no treatment for us. And now we have been able to identify several genetic mutations which are actually responsible for the occurrence of these diseases. So if at some point we can actually splice these genes, take off that particular mutant gene, perhaps this might result in a lasting relief, or we can have some medicines being given which can actually suppress those genes. That will be a huge potential which can be done. The other important neuro-oncology, glioblastoma is probably the most malignant tumor known to the brain. And the problem with glioblastoma is that no medicine actually acts on glioblastoma, and a person who gets a medicine against glioblastoma is going to definitely win a Nobel Prize. There's a person called Roger Stupp in Geneva. He was a co-resident with me. He has brought out one medicine which is known as temozolomide. Now, the only advantage which temozolomide gives is a 3-month advantage in a particular group of patients. And the reason why no medicine acts is because the tumor continuously changes its antigenic character. So even if you get autoimmunity against it or any medicine against it, then it immediately changes its character and becomes a new one. That's a very important thing. Then there is very important area is attenuated viruses. Say for example, if you can directly implant attenuated viruses or radioisotopes in various parts of the body, then they can selectively give radiation, they can give medicine, they can give drugs to various parts of the body. So that's a very important area that you can work on it. Then there is another thing, chip. So this human on a chip, say for example, a diagnostic chip, supposing you have a small chip and just put a drop of blood and you can get all the investigations done. That is 3D printing is a very important area. You can actually make the organ. There is no need for organ transplant from another human being. What you can do is create 3-dimensionally an organ, and you can do all the pharmacology tests, toxicology tests, and the same organ can actually be transplanted into the human body and can make it function. So that's a very important area that can do it. Then nanotechnology is that, take small pills, which are nano pills. We can ingest those pills. They go to various parts. You can actually guide them along electronically. They act at the site and can work on that. And then there is one important area is artificial intelligence is a huge thing, because supposing you have a radiological diagnosis or a pathological diagnosis, then that can easily be done using artificial intelligence. And the final point I'd like to make is that this is now going on research, that using a small electronic gun, you can put chips at different points. So there is a paralyzed part of the limb, you put a chip, and that stimulates the nerve, and that causes physiotherapy, neurorehabilitation. So that's a very important area that we can focus on. So there are several areas in medicine where nanotechnology, plant medicine, and various other aspects can work. Thank you.
Thank you, Dr. Sanjay Behari. Sir, Dr. T.V. Nair, please.
Thank you. Thank you. Let me, at the outset, thank Professor Mohanan for making me here. And I'm extremely happy to attend this conference, international conference. Although I used to visit this campus quite often, especially talking to the young biotechnology students over here. And this conference, I'm sure, is organized very well. And in the beginning, when Dr. Sanjay Behari was talking, on an overview of biotechnology, what biotechnology is, I told him that, he should stand corrected, that he was the apt person to give a gist of the entire developments of biotechnology in a very limited time. There is nothing much for us to talk in a general way. But as a person who has been involved in biotechnology for the past several decades, I feel that there are so much of things happening in the country and abroad in the biotechnology field. As you all understand, that biotechnology is one which reaches the society. Any product or process that's being developed through a biotechnology intervention should go to the mass. Dr. Pandit was talking about the GM crops. Of course, we know that it has been developed. BT cotton was developed here. Several other crops have been developed with several kinds of traits. But unfortunately, none of this, except the BT, did reach the farmer's field. Now we have the GM mustard being given for limited trial, but I do not know how far that's going. But then one area that is promising is the CRISPR technology, because that involves only editing the genome, not just borrowing something from elsewhere and putting it. Maybe because there is a mudslinging happening in the area of biotechnology, especially in genetic modifications, that people say that we are practicing a kind of witchcraft, or it's something which is going to harm the entire biodiversity. I do not know how far that should be taken into consideration, especially the NGOs would be speaking loud where the scientists are silent and we don't speak. I've been talking at many for advocating the advantages of this, but then of course I'm a small man to speak about it. But understand that the newer technologies that are coming in biotechnology, especially when you take, as a product or process, it definitely reaches the society. And that should be the vision of biotechnology, because it should be a science which should not be practiced in a compartmental level. It's an truly interdisciplinary science. We have spectrum of people sitting here from different disciplines, but then the convergence is to make something which would go to the society. If you practice at the lab and it doesn't reach the society, that means, it's just a basic science. Biotechnology, as I understand, it should be an applied science, which should have a lot of application value. We have several areas like what Dr. Sanjay has already said to you. Especially, I was with the Kerala Biotechnology Commission, one of the co-sponsors of this. While I was there, Dr. Sharika is there. We have started this Institute of Advanced Virology. It is a time when we had the pandemic. The government of Kerala had a great vision that we should also have the second virology institute in the country, because we had to send all the samples to Pune, and National Virology Institute, Pune. But they wanted to have it. So we all put our heads together, and within a record time we have established, and it's functioning very well. But why I'm saying is that we need to be prepared. For example, not just for a pandemic when it comes to us. We need to be prepared. We need to have enough gadgets and machineries to combat this. Viruses are going to come. Pandemics are going to come. Are we prepared? We don't have a vaccine for any of the viral diseases. We have only for COVID that was necessitated by the pressure that we had gone for the Covaxin and Covishield, which has gone into the market. I'm sure that Dr. Mohanan was one of the experts in the committee to approve that out of way that they had approved it without a clinical trial. So normally it should go for a clinical trial. It takes about 10 to 15 years for a drug or a vaccine to be perfected. But then that was needed, and then it has gone to the mass. And many of us are alive, or maybe that we are now in a position to fight COVID mainly because we have taken the vaccines. So likewise, there are so many things. You need to have a effective diagnosis. You know that how biotechnology has helped in the diagnostic procedures. At the nick of the time, even if you see in many of those institutions, in many of the laboratories, that we have testing facilities available. RT-PCR has become a term which is used classically by everyone. No one was knowing what RT-PCR. Now everyone knows what RT-PCR is. So that means we need to have more of diagnostics. Before a pandemic comes in, before a problem comes in, we will have to have several measures by which we can combat it. Preparedness, prevention, as well as we should go for managing the situations. So if you can do that, and as biotechnology, we have a response to do that. We have several viral diseases, we don't have a drug. For many of the viral diseases, we don't have drug. We don't have a drug for COVID. So like many other virus diseases, we don't have a drug. So how will you? But one of the problems with the drug development is that it would take several years for making it perfect. But then once you make it, the pandemic is gone or the virus is gone. So maybe that no industry, no pharmaceutical company is coming forward to make a drug. So there are so much of opportunities available. The young mind should say that it is not a science which should be practiced in the labs. It should be taken outside. You should have a vision. You should have a kind of dream to make things happen which would definitely reach the society. So as a biotechnologist, I have been practicing, I have been telling people that unless it is taken and translated into a process or a product, we need to have clean water, we need to have good food, we need to have nutritious food, and we need to have good drugs. All these, I'm sure that biotechnology is the process which can develop that. Not only that, to the environment, to the agricultural scientists, you know that the plants are facing so much of abiotic and biotic stresses. Abiotic stresses that, as you know, that the drought, the salinity, all these are affecting the crop yield to a greater extent. So we should equip the plants, crops with that kind of machinery by they can either tolerate or withstand the salinity, drought conditions like that. In biotic ones, you know that we have not much of diseases, the viral diseases in plants, which causes a lot of menace. We have a lot of fungal diseases, and all these are to be, they're all about biotic pressures that goes into the system. We have several measures. BT is something where you have that kind of resistance being developed into the plant. So if the plant is defending, why should you bother that whether some new gene from a bacterium has gone to it? So maybe that as biotechnologists, it is also your responsibility to get to the society and tell them that what exactly you're doing. Don't do things in a compartmentalized way. Everything should be open and people should understand. Probably that was one of the reasons that people did not understand biotechnology properly and take it to the society. So maybe that you need to specifically practice it and practice it with an intention that you would contribute something to society. So you could become an entrepreneur, you could make a startup, or you could align with an industry and make products developed which would go for society. For the timing, I'll stop here because this is a general kind of discussion. So some of those major problems that is caused by biotechnology and how we can overcome that and, suggest remedial measures, at least in this kind of forum, if we can suggest something, not only to the agricultural university sector, but to the government of Kerala as well as government of India, that would be something. So we should put points as a recommendation for making this. Thank you.
Thank you, Dr. G.K. Nair. GM Nair. Dr. Achut, please.
I would first of all like to congratulate the wisdom of the organizers in asking us to speak about biotechnology in 2047. That is quite easy to talk about. Nobody is going to come after us and check whether this would be realized. Had it been any short time frame, we would have been in trouble. I would like to preface answering the question that the moderator has raised by mentioning that life is being looked upon or being looked at through different windows. The window of, traditionally it was the window of biology, later biochemistry, then biophysics, especially the DNA structure elucidation and all is an achievement of physics, if you could agree. And so bioinformatics cannot be far behind. It is a window to look at life by studying the information that seems to be embedded, written chemically on the rugs of the ladder of the DNA. And there is a very visible interdisciplinarity in the whole field that has emerged during the last 100 years. I sometimes feel, the first thing that happened in the computer field, taking inspiration from the computers from biology, is that computer scientists produced a virus, in the computer virus. And today we are talking of CRISPR. In some way it reminds me of gene editing, reminds me of in your Word document you are cutting and pasting, the idea goes to the other side. Now, when I say biochemistry, biophysics, biology and bioinformatics, I think biotechnology does not fall into that line. It is in a different vertical. So far, biology has been a science trying to analyze the wonderful phenomena of life, but today biotechnology is a maker. It makes things which do not exist in nature, modified organisms or modified crops. So that is a very important development in this field, where I think that the whole ethos of the subject of life science in one corner has changed to something like engineering, where you are making things. One of the most wonderful things that I can think of is the lab meat the non-violent meat that is being now coming to markets. And then of course we have been talking about BT cotton and then also terminator seeds. Now the thing is that we are now getting more levers on life whereby they can be modified to suit our greed and our business interest. That is what is happening in the field of biotechnology from a slightly political viewpoint. I've been asked what are my comments on the issues that are in the field. I would say all my eminent fellow panelists have mentioned environmental, ethical, and I would add political. There would be crops which are introduced to cause damage to a nation or to soil or to a place. There are viruses which are engineered and introduced as a means of warfare. So these are all issues that could arise in future. And I was also asked what is the impact of development of biotechnology in my field. I will cite one example. Because biotechnology has arisen, there is a need to design organisms, and when new organisms are to be produced, whether it is plant or virus or whatever, or bacteria, modified bacteria, which is very popular, there is a need to do a soft design of the organism, and this area, which is the general area which is called synthetic biology, requires mathematical modeling and computing to test out things, to produce a very strong hypothesis that we can go into the wet lab work. And so this area is growing. I would refer to the synthetic biology workbench software which bioinformatics has produced. This has come out of a need by biotechnology, development of biotechnology. We even have a small programming language called synthetic biology markup language, which would mean that on the computer screen you can take genomic components and assemble it by click and drag and see if the gene is capable of expressing, so that you have some confidence before going into modifying a bacteria or any other organism of your choice. So I think it definitely, biotechnology definitely has a very great impact on bioinformatics, or rather bioinformatics becomes more relevant because there is biotechnology. This is what I feel. I will add more points in the next round of discussion.
Thank you. And Dr. Soni, please.
Good evening to all. I'm honored to be here in this, among this great panelists. And about the question on the advances in biotechnology, I would say that I think I will start from my MSc days. I was in the first batch of biotechnology in Kerala, from Cochin University of Science and Technology. So at that time, nobody knew about biotechnology. Actually, what is biotechnology? That term was very new to all. And we also suffered. Actually, we didn't have anybody to teach and to show how the biotechnology was. No labs and nothing. And after a long time, I witnessed, that is after a long period, I witnessed the advances in biotechnology. It was actually a great leap to different types of techniques and tools which can be used in different aspects of life. You can see that biotechnology is not actually, it's an independent science. it is extended to the different fields. It can be used, it may be except in the social sciences. But now I see that the social science department is also working with us. So that advances, if you see, you can see that it started with the very small techniques and all, and after that it went to that very debatable type of techniques like GM technology, transgenics. And when that type of technology evolved, that was a time when people started actually staring at biotechnology and looking at biotechnology in a different way. The dimensions also changed, the viewpoints also changed. So at that time, many type of opponents also came, the public offense and all. So at that time, so we can see that when the opposition from the people come, so you can see that it is actually going forward. It is actually growing. So we have seen how the GM technology worked. And we could see the many different GM crops developed. And after that, it is also still, it is growing. And at the same time, we could also see the different advancements like RNAi technology, where again people thought of having less toxic, having lesser issues compared to the transgenic technology. Then again, we saw the advancements in the field of nanobiotechnology, where we could see still it is a growing science actually, how the biotechnology the nanotechnology can be incorporated with biotechnology. So you can see it is growing like anything. So you can see its use in the different aspects, like from the pharmaceutical industry, the medicine, and in agriculture also. So that is still growing. So again, people don't know how to regulate this type of, because we don't have a clear-cut idea about or to formulate some guidelines for the nanobiotechnology in different way. Because it is an integrated type of thing, different departments are involved, different departments, and a lot of things are involved in nanotechnology. Then, as our sir was telling, we can see also the coming of computer into biotechnology. Because we didn't have any idea that computer can play a role in biotechnology. We are using computer for storing the files and all. But these statisticians, mathematicians, and the computer people, they came to actually to help us. So that happened during the Human Genome Project. So it was a time when actually needed the help of the computer people to store such a huge, that big data. And we can see that it has become an indispensable part of biotechnology. So you cannot find that is the biotechnology growing without the help of bioinformatics now. Now, by latest, we can see the emergence of CRISPR technology, genome editing. And genome editing, actually, it is revolutionized biotechnology, because it has become a very precise tool. So compared to the other GM technologies, you can see that it's very precise. There is no transfer of the genes in that. So it become, the people started thinking positively to this technology rather towards that GM technology, because now they are sure that it is not actually a GM. It is not, there is no foreign gene in that. So because BT was a terror to the people, to the society. Now it is growing actually. So you can see that the CRISPR is creating many type of, that is, it is being employed to create, especially that is in the case of agriculture, you can see for the crop improvement programs, how it is being used for developing the stress tolerant crops, for the quality traits. So for many things it has been used. So that is how I see the growth of advances in the field of biotechnology. And again, to that question about what is the limitation or So what I see is, like these technologies when they evolve, because it is from the biology side, people will have that fear about that. So like that, there are different points to be discussed in the case of the transgenic and all sort of organisms which are modified with different tools, whatever it is, whether it is transgenic or cisgenic or any that's CRISPR also. So we have to see the aspects like the social aspects, the legal aspects, ethical aspects. So there are several things to be considered. Even the environmental aspects are also there. So we have to see that as a scientist, we have the responsibility to see that this science is safe to the people. So you can also see that we have stringent regulation of the GM crops, whatever technology we use. So still you can see that it is not accepted. So in India you can see the first GM crop was introduced in 2002, but still we cannot see any GM crop grown in our or commercialized in India. So what is happening actually? So this is a point of discussion actually we should have. So what I feel is the public trust is the most important factor. They should trust our technology. How is it possible then? So that is the most important challenge the biotechnologists have, apart from the other things, because that can be solved, like the legal aspects and environmental aspects, socio-economic aspects. We can, to some extent, we can solve, I think. That is my belief, because there are ways to solve that. But what about the trust? Trust has to be built. So that is the major challenge, the most important challenge we have. So that means the responsibility is with the scientist. So we should see that our technique is the soundness of the techniques we are developing, how it is going to influence the nature, how it is going to have some interaction with the nature, the environment, the people around, the health of the people. So with this, we have to change the mindset of the people around us. So as the many panelists were discussing about the limitation as the time we are taking for releasing the crops and releasing of any type of product we have. So what is that lag? So that lag is because of these issues. So still in India, we are not able to release any of the GM crops. That doesn't mean that we are not doing the work. At many labs in India, they are at least the latest technology, the CRISPR-Cas, been tried by many people, many scientists. Even in Kerala Agriculture University, we are using the CRISPR technology. But after that, what to do with that? So for any type of genetically modified crops, especially the genetically modified crops, compared to the other products, we can see that it is the lifetime work of a scientist. But still the scientist will be going back without seeing that product outside. So we have to think about that. Where are we? What is to be changed? So how to solve this problem? So that has to be discussed. Thank you.
Thank you, Dr. Soni. So we'll come back to these questions very soon. I think a couple of points raised by common members. Let us listen to Dr. Rajiv Nair.
Thank you. So first of all, let me also thank the organizers for inviting me, and I'm happy to share the dice with very elite panel of biotechnologists who are experts in their field. So almost all areas have been discussed, probably except energy, which I am working on, happen to be working on. So when you look at how biotechnology has influenced energy research, or what is happening, or advancements in energy, I can think of almost every area, starting from whatever our panelists have mentioned about gene editing to even waste water management. Everything has, is coming in the area which I'm working on. So we have been seeing developments to begin with, I also would like to say that, whenever we are talking about, energy, the need for a sustainable energy, renewable energy, we are also talking about how to replace the fossil fuels and how, if you don't control the global warming and keeping the global temperatures below, should not rise above 2 degrees Celsius in this world, where if that happens, you're on a non-returnable path. So there is a need for shifting to this, renewable sources of energy. And tomorrow's crude is biomass. Probably when you look at in a renewable source of energy, we need to actually utilize the biomass for this, converting that biomass into different forms of energy, which can be through any route. But of course, biotechnology plays a very important role there. If the biomass would have been easily degraded in nature, that would have probably there would have been no life on Earth. So biomass is recalcitrant because of a reason, and it is very difficult to degrade the biomass. So there are, in my area which I am working on, there has been efforts for making this biomass degradation pretty simple or easy to work with. So there has been studies on how to increase the yield of cellulases, the enzymes which degrade the biomass, more efficient, and how the microorganisms which secrete the cellulase can be engineered so that we get high titers of cellulase and we can actually degrade the biomass more efficiently. So all those technologies that we have mentioned, gene editing, everything has been applied here, even currently being applied here. So there has been advancements which have resulted in better enzymes, engineered enzymes. We have been looking at, again, protein modeling. You're talking about artificial intelligence. I mean, over the past few years, we have been hearing a lot about, AlphaFold, which is probably one of the best, artificial intelligence-based engines for modeling the protein. So if you model a protein, especially cellulases in my context, to get its structure engineered so that it performs better. So you have a lot of possibilities coming up, lining up. And for certain enzymes which we don't know what is the structure about. So certain clues on that will definitely help this kind of research. And then of course, you have been also probably have read about Craig Venter's creation of an artificial organism, an engineered microorganism with a whole DNA coming from another organism, put it in the cell of a different bacterium. And the whole purpose, if you look back and read the paper, was again to make this organism to produce energy molecules, energy carriers. So that was his original intention. So these kind of research, I mean, everything has been applied towards developing better organisms, better systems to convert the biomass and to generate energy molecules. And there has been also different kinds of attempts like making engineered plants like sugarcane. Sugarcane has been engineered to produce oil inside the cane. So this is called oil cane. And the advantage is that sugarcane is anyway cultivated. So the advantage with engineering something onto an agricultural crop is because you don't have to change the land usage. And land usage changes are very, very, it creates a lot of penalty for the carbon, emissions, as well as, to curb the carbon, we should minimize the land usage changes. No, it doesn't change the taste of the cane. You're actually making the cane to produce more oil. So after crushing, you're also getting oil as another product. Anyway, you're getting the cellulose, you're getting the juice. Along with that, you also get the oil. So when you talk about creating oil using a microalgal cultivation, the biggest problem is, from 1 liter of water, you're getting only close to something like 1 gram dry biomass of algae, which is never going to take off. Because on theory, you can tell that, this is all very wonderful, it is exciting, but in reality, I think one of the panelists already mentioned about how it has to go to the industry. So in industrial perspective, you also need to look at what is the feasibility of these things. So certain things are interesting. Tomorrow that becomes a reality, but as of now, we need to look at what is the feasible things. So there are multiple different areas, carbon dioxide capture, and converting that carbon dioxide to some sort of energy. And also, even, I think Achyut Shankar sir will agree, there is a lot of applications of artificial intelligence, machine learning coming into that. We are also applying some of these things. Actually, one of his students is in my group now. He has developed a program for an algorithm which can feed in FTR data and actually output the composition of the biomass, which actually takes 2 days to the estimation. But in fact, with this, you can actually feed in the data, half an hour, you will get the composition. So this, these kind of developments, coupling biotechnology with, different other fields has to happen. Of course, biotechnology is an interdisciplinary subject, but of course, a lot of things are coming in different areas, different fields. So anyway, the time, I think, is, probably.
I think there's a lot of interesting examples on synthetic biology and engineering coming up here. So it would be more exciting for Dr. Achyut as well to explore these options. And so, when we talked about engineered genes or engineering organisms, engineering viruses and all, so one of the concerns looking at, say, for example, genetics, with more and more information coming about the genomes and human genome projects. So how do we address the problems of, say, congenital disorders, typically like cardiac disorders, which are congenital in nature, which are gene-linked disorders, which are coming up. So are there any solutions? Have we gone any good way ahead in these directions? My question is specific to Dr. Thomas, and probably Sanjay can complement after that.
During the last 20 years, many mutations in genes have been identified, resulting in congenital heart disease. The problem is that many of them are de novo mutations. So it's relatively difficult also genetic counseling to predict and then also, of course, specifically intervene, even if you would have a precise method in correcting such a disease. Many of these de novo mutations, they appear in the patient, in the kid, in the baby for the very first time. Then, of course, very difficult to do anything. You just can handle what happens. Of course, in principle, one could think also about much better screening early on to do early interventions, but the main problem is de novo mutations. And a second thing, which is probably not so well known for most of you, actually the highest risk factor for congenital heart disease, what do you think is that? Obesity of the mother. That's actually the highest single risk factor for developing congenital heart disease. So it's actually not known how this happens. A lot of hypotheses, changes in lipid modifications of sonic hedgehog, many hypotheses, but it's not clear at all. So I'm afraid a really good solution is still on the horizon, still probably beyond the horizon. So I think really early diagnostics develop methods, and I think some of them for trisomy 21, it's possible already to detect trisomy in the blood of the mother. Very simple. To develop better screening techniques, also allowing early interventions. But I'm pretty sure this will not cope for all types of these de novo mutations. Sometimes you have even siblings. One has a congenital heart disease, the other not, because one attracted a somatic mutation during embryonic development, and the other sibling or twin, I wanted to say, not. So of course that's very difficult to handle prospectively.
So also there were a lot of questions up in the WhatsApp on from the audience, basically on how the metabolic disorders or the high lipid levels. So a lot of questions on how is coconut oil good for health. What type of fats are good, say monosaturated, polyunsaturated, or so Dr. Sanjay, do you want to add something on to this line on what are these lifestyle diseases, and are there anything in perspective?
Professor Salim Yusuf had come the other day to Sri Chitra, and he has done a multi-country trial which actually involves more than 100 countries. where he explored the non-communicable lifestyle diseases and what are the factors which are actually affecting them. So he found that one is, of course, the age of the patient. The higher the age, the more the incidence of atherosclerosis, the more the blockage. The second is low density lipoproteins. The third very interesting point that he mentioned was that carbohydrate diet, in contradiction to lipid diet, we used to think that lipid diet is responsible for deposition of atherosclerosis, but the real harmful product is carbohydrates. And a very low carbohydrate diet has a significant effect in decreasing the disease of heart. Then the fourth important thing is diabetes, hypertension, and even by decreasing blood pressure by 10 millimeters of mercury, you are significantly, almost 60% decreasing the chances of some problem occurring. And diabetes is another very important factor which can result in this. Then a subclinical heart attack. Sometimes, what happens is that there is a little bit of chest pain, you don't really look at it, and that causes an issue. These are some of the factors which are, some of the minor factors include sleep deprivation. So significant change of sleep patterns and sleep deprivation is an important factor. These are some of the lifestyle diseases which can actually affect the heart. But there is another aspect to it. When we talk about congenital heart diseases, although this is not an aspect of biotechnology, but there are several interventions which, when done on time, really help. Like, for example, a one-day-old child or a 1-week-old child we operate in Sri Chitra, and the advantage of that is that before the child develops a neuropsychological trauma, you can actually intervene, take the thing before the heart muscles becomes weak. You can actually intervene in this child. The second very important thing is, supposing somebody due to the heart disease develop a congestive heart failure, then the advantage is that you can do a biventricular pacing using an endovascular technique. And we are also making those stents, we are making those guide wires and everything. So once a biventricular pacing is done, you have time for intervention prior to this is done. And the final point that I'd like to mention is a very interesting point, that what we do is we harvest cadaveric valves. And supposing somebody has atrial regurgitation or mitral regurgitation, and we harvest cadaveric valves, keep them in a biobank. We now have nearly 300 cadaveric heart valves. And the interesting thing is, when you actually implant them onto the heart and stitch that up, there is no immunological reaction to them. Within the heart, this is a very strange thing. You don't have to give any immunosuppressants to decrease the autoimmunity against those sutured hearts. So these are some very interesting possibilities of research and work.
So we heard about, say, going slow, and the progress has been slow in the case of genetically modified crops as well as for gene editing and application. So if you look at the central government, for example, one of the major funds in biotechnology or crop science has been, one is for the millets year this year, and the second one is developing and establishing a indigenous gene editing facility or center nationally. So there has also been emphasis on how do we promote organic crops. On the other side, coming with genetically modified crops for better yield and a lot of other advantages. So my question was specifically to Dr. G.M. Nair. So how do we take a stand? We need to speed up faster. Say some of our neighboring countries have gone way ahead, like Bangladesh, for example, the GM crops. So they have been one of the successful models in developing GM crops and getting a very high yield and productivity. So what are your viewpoints on, what are the suggestions on how do we speed up, and what are the policies to adopt in this direction?
As you rightly said, there has to be measures by which we can speed up this. Of course, you know that any kind of modification that you do onto the crops, especially through genetic modification, is going to perennially affect the generation. Now, when you do that, you need to be very specific what exactly you want to do. you have to convince the authorities where the approval is to be derived. So you need to look at those things. Otherwise, what will happen is that many of the things are happening are in the lab scale. It's not allowed to go into the farmer's field or in an experiment. A classical example is one GM crop developed in rubber by the Rubber Research Institute of India. It's for tapping panel dryness. That's one of the major problems that you have. It doesn't affect anything. It's only that, superoxide dismutase and that kind of thing, manipulation that they did, by which that can withstand the dryness in the course. But that itself was not allowed to go into. So what I would say that rather than looking at that, and you are sure that you're going to get the clearance for that, in that case, why can't we take up such technology like gene editing, the CRISPR-Cas technology? You are not borrowing anything from the other source. You are making an addition on a precision way to what modifications you will be doing. That would be an approach which should be promoted, and I'm sure that you never thought in 2012 or so, you never thought that CRISPR is going to make that kind of a revolutionary change. And maybe rather than wasting time on genetic modifications this way, and if you look at that, and then I'm sure if there could be much easy recognition and approvals that can come. As you know that all these are very expensive programs, and then of course they are input intensive. Unless it reaches the society, it becomes a lab scale work only. Biotechnology is not meant for that. Biotechnology should go as a product or a process or improved organism. Nutritional quality is improved, its defense mechanism have to be improved. For all that, I'm sure the answer is CRISPR-Cas9 technology. I'm sure that it
So back to Dr. Soni, when she mentioned about gene editing as a tool for way ahead. In fact, if you look at a decade back when people were looking at transgenics, it's almost like posting a letter without an address, or I mean, in terms of gene editing. So we have gone specific to which gene has to be edited, which are the set of, whether it is SN1 or SN2, we can have editing a total gene, or where it has to be placed, how many copies have to be made. So these have become very specific now. So what else need to be enabled in this field? Of course, proceed with question is always a nod to avoid all other factors. But from a pure biotechnology perspective, even now there are restrictions on gene editing tools to be used, on number of mutations you can make, etc. But what could be a policy way ahead so that we can speed up with caution?
Okay. As far as that regulations are concerned, in India, we have the regulations for all these technologies. And for genome editing, actually, it is exempted from the type of stringent regulations, at least for the 2 types of mutations they are making. That is SGN1 and 2. But 3 they are not avoiding, just to go through that. So that when you see that type of technology, so you can see that if you are making use of the precise tools, because CRISPR-Cas9 is the very mostly used one. So now we can see that even the other technologies, like the most evolved ones, like base editing and prime editing, has come to do this job very precisely. So in that case, we can go for the avoiding. We can avoid this type of stringent regulations in the gene edited plants. And about that, what else? That is quicker to make it more quicker. So we have to include the type of monitoring. Monitoring is very much needed in this aspect. So that is what it is missing in the case of the GM crops also. Because we have a set of rules which is to be followed for all sort of genetically modified organisms. But when you go through that different steps, you can see that it reaches the last step, but still it is not approved. So that means some sort of monitoring is required. So that monitoring can include the different aspects, the people from the society, that the layman, and then the technologists who can monitor actually what type of the thing is actually we need. if it is edited one is correct or any off targets. So there are technologies now to avoid all these type of off targets, because that was the question in the case of the genome editing. So that is, there are now, the technologies evolved to avoid all these type of that off targets and all. So what I would say is, actually the technology is clean, only the monitoring is required. So we have to say that with the different type of products it is formed, where to monitor, how to monitor, and what type of techniques to be used to see that whether it is the one we really want.
So in the inaugural session, Dr. Sanjay quickly mentioned about the immunospecificity and immunotherapy practices right now. So could you shine a little bit more light on to, like, how biotechnology can be a game changer in
So there are different techniques of immunotherapy. One could be immunomodulation. So for example, there are several medicines which can actually cause immunomodulation, which can actually increase the immunity against any organism which attacks it. So that is one way of immunomodulation. Then there are certain drugs which can be given. For example, methotrexate is one drug which can be given. And once you give that drug, it can actually enhance the immunity, and which is a focused immunity against something. The third very important way in which immunity can be worked on is giving some therapy, immunotherapy. Like for example, you have an anti-rabies vaccine. Now rabies, if a dog bite, if you give a vaccine following the dog bite, it doesn't work. So at that time, if you give immunoglobulins, then that actually works till the vaccine actually takes effect. So that's a very important aspect of immunotherapy. Then the fourth way is immunosuppression. So that if there is a person who has some kind of organ transplant, then you don't want the body's own immunity to act against organ transplant. So in that case, you want to suppress the autoimmunity. So that is another way of doing it. And the fifth way you can actually modify immunity is bypassing immunity. For example, in the brain, there is a blood-brain barrier. So because there is a blood-brain barrier, so what happens is that the white blood cells and microglia, actually they don't go to the brain. So if you want some tumor to be destroyed within the brain, you can stereotactically implant an attenuated virus or attenuated ricin poison into the brain cells, or a radiation source into the brain tumor, and that can act locally to cause local effects. So there are several ways in which immunity can be modulated to work for you.
Right. So I think Dr. Achut was trying to add something here in this topic. So very specific on personalized medicine, for example, like personalized medicine, there is a lot of studies in the Western world now looking at drugs, the drug reactions, and the database being created about the response of each and very specific. So pharmacogenomics has been one of those very hot areas right now. So can you comment on how much of pharmacogenomic knowledge is being exploited in our regular treatment conditions or disease
Thank
state?
you.
I would like to preface it generally with how the knowledge in bioinformatics has contributed to the transformation in this related area. Generally, if you look at modern life science, what you could call as new biology, just like we run to Google or now ChatGPT, a study of life at molecular level starts with reading the genomic information and then rushing to the website called BLAST. Everything begins there. In fact, I could cite a very good example. For decades we have debated on Darwin's theory and whether it is acceptable or not. But today, if one of the young people here raise a question whether Darwin's theory is acceptable, can you prove it? Then we don't need any debate. All I need is a drop of your blood, and I would have a small segment of your genome sequence in a day, and then come and paste it into BLAST. And however beautiful you think you are, the second hit is the orangutan and the third is the chimpanzee. And do we need to debate any further? There is no debate there. Actually, from a very subjective debate of theory of evolution, we have reached a very objective kind of a measurement of the kind of evolution. Now, I would like to mention that computer-aided drug design, molecular docking softwares are working behind the curtain to really study the effect of a small ligand molecule on a target molecule in the disease pathway. And coming to one more example of personalization, Professor Sanjay Behari knows about this. As to what happens in his own institute, I understand is that the exome sequence is made and the drug reaction is actually studied from some markers, which could be local also or could be de novo also. And from that, the drug administration is actually decided. So I think at every level, the level of designing drug molecules to the level of administering it, kind of personalization is possible with this genetic information. This is what I quickly would like to react on.
Yeah, Dr. Thomas, yeah.
To add one to thoughts. Actually, cancer, I mean, the German Cancer Center in Heidelberg, they are now sequencing all tumor patients, trying to extract new targets, and sometimes you indeed find molecules that you can directly use. But the second problem then, of course, emerges, you alluded to that, tumors are moving targets, and quite often you achieve a certain remission, and then you have to go the whole circle again, try to find new molecules, and I think there bioinformatics, also structural biology predictions to rather rapidly come up with new potential targets. I think this is an area that will flourish in the next years. And maybe also due to better understanding of biological processes, one can also select better which is a promising target instead of just dealing with something that's more or less an epiphenomenon. Second point that I wanted to make is regarding inflammation, which I think is really a double-sided sword. I mean, for regeneration of many organs, you need inflammation, so during the initial stages. But later on, it becomes detrimental, so chronic inflammation is usually always bad. But that also means that you have to introduce a new dimension, the timeline, when to manipulate what. So if you do that at an inappropriate time point, well, then you might do more harm than good. And I think that's also, also applies for many other biological processes, that a precise timing might be really decisive, and one needs to be careful in figuring out what the right type of approach is there to do manipulations. But in terms of precision medicine, I think particularly in tumor medicine, things have really advanced. It's just this Darwin-driven evolution of tumors that is the major problem. If they would be static, I think we would have cured cancer, but it isn't a static disease.
Interesting. So I couldn't restrict myself from asking one question to Dr. Sagar. So this was more related to the problem of climate change. So climate change, so when we talk about changing weathers, high temperatures, talk about drought-resistant varieties, talk about high temperature with crops with high yield, etc. So what insight into molecular biology can address these changes in ecologies? Or again, the type of pests that are attacking the plants change with the different climatic conditions. So do you have any insight into this knowledge about, say, the changes in weather can help in better growth?
Yes, can you hear? Yeah. So I think we are actually getting into the climate smart agriculture simply because the climate change pressure is enormous at the moment. If you want to just take this example which you mentioned of pests, then it all boils down to something called as evolutionary arms race. Or We also call it as a biochemical warfare. At any given time, a plant and its enemy, whether it is insect or it is a pathogen, they are in a constant battle with each other for some kind of one-upmanship. You know, if you see a susceptible plant, that means in the course of evolution, a particular insect or that pathogen is at this moment of time is higher on the evolutionary scale. It has developed some kind of resistance against the plant's character. And so is true with the abiotic stresses when it comes to the climate change. Now the whole question is how quickly the climate change is going to take over. When we talk about climate change, many times we confuse between the climate change, for example, the Pleistocene glaciation that comes under climate change, versus the current anthropogenic changes which are really expediting the climate change. We really don't know whether these anthropogenic changes and how much will they add to the climate change. So we are actually, there are a lot of scientists who are working on understanding the responses of plants. And in this particular case, not the crop plants, the wild plants from various different forests, may they be temperate, may they be tropical, and how they are responding to the climate change by their ecophysiology. So this is totally a different dimension than what the agricultural scientists deal with. And from these ecophysiology studies, and then narrowing down to the genomic causes of ecophysiological changes, they are trying to find out what exactly enables the plant to cope up with the climate change. So I think, yes, genomic studies are going to give us great insights into this, but we need to do more such studies, especially if you are talking about the climate change. We must little bit, divert away from the crop systems and try to study the non-model systems which are actually at the defense line, the front line of the climate change battle. So these are the species which are going to give us, our candidate genes to be incorporated in the crops later on for making the climate smart crops and so on. So I think, yes, we should go on for the genomic studies in such non-model systems. And we already had a lot of discussion on GMOs and other type of biotechnology issues. But when it comes to, again, making your crops climate smart, you will have to go to the, gene modification kind of thing. We must remember, I'm not an advocate of gene modification or GMOs, anything. It's just a very rational view as an ecologist and biotechnologist, both the things coming together. That's a rare combination, by the way. But whom are we fooling? Why are we flattering ourselves? We are the species who, in spite of knowing that smoking is injurious to health of the smoker as well as the bystander, we smoke. We are the species, we know that destroying the forests, deforestation, is literally suicidal to this mankind and this planet. And still we are overzealously doing that with a rapid pace. So when it comes to, facing the increasing population pressure, we are adapting to everything. And so we are going to adapt to the GMOs. So they are sooner or later going to be a part of our life. I don't think we can resist it for a longer time. be it a climate change smart species or be it productive agriculture, etc.
In fact, with our successful Chandrayans and Mars missions coming up, we might really have to look at the biotechnology for the space, which could be one of the upcoming areas where we might need all of these tools in hand. So I think we are running out of time. We have already crossed our limits, but there are a huge list of questions that has already come from the audience. But I would really want to take a couple of questions from people here around.
Just 1 second,
Yeah,
can I talk
sure.
a little bit about space medicine?
Yeah, sure, please.
A very interesting point which many of us don't realize is that there is a short space walk and there is a long space walk. Say for example, if somebody is there for only 3-4 days and comes back, there's no problem. But anything longer than that, gravity has its effect. And actually I was editor of one section of the space medicine book, so then it's available online. The interesting thing is that because of gravity increase, the fluid does not come to the feet. It stays in the brain, and that leads to increased intracranial pressure. And this increased intracranial pressure actually manifests into the eyes and causes visual loss. So if somebody has been in space for a long time, it causes visual loss and because of papilledema and secondary optic atrophy. The second very important aspect which happens is that there is a huge loss of muscle mass because gravity actually strengthens the muscle. So every person who's in space, say space station, they have to actually exercise several times a day. They have to exercise for 3 to 4 hours a day, otherwise they are losing their muscle mass. And even when they come down from space, after staying in space for about a year or so, they have to be carried because they can't walk. The third very important thing is that there is a subtle radiation which comes. So you have the incidence of the Kelly brothers. They were identical twins. So the brother who stayed back on the ground and the brother who went into space and stayed there for 1 year, there was a genetic mutation in that brother, so their genetic composition changed. The fourth is important thing is decalcification. The bones rapidly lose their calcium, so there is going to be a kyphotic deformity. So when people say that a long space flight is possible, it has huge reservations, and space has huge potential for research and for applying experimental medicine into it.
Thank you.
I had one point to add if you're asking the questions.
No, please go
No,
ahead.
if you're asking from the
No,
questions.
let them, can you circulate the mic by the time Dr. GM
No,
the mic is already there?
my
okay, please go ahead.
point is away from what is being discussed. It is about quality biotechnology education. Because we are making products, we should go to the society and do something, isn't it? Are we equipped them that way to go to an industry or to make products by themselves so that they can translate and make products? Now the problem that concerns is that we have no industry sponsorship in many of the programs. Biotechnology, especially in the pharma sector, the biotechnology, if it has to reach, it can reach only through a pharma company, okay? Now, we should ask the questions to the students. How are they equipped? Are they skilled? Are they practicing biotechnologies or are they theoretical biotechnology? Practicing biotechnologies will come only when they're skilled.
Excellent question. We already have this question here. Are we industry ready? What type of curriculums or changes in
Shape
curriculum would you
So involve
expect
industry,
for the
and I'm sure
biotechnology?
that most of the national agencies are going to stop support for research.
Yep.
And especially with the amalgamation of all the agencies together, there will be a lot of problems. CSIR also has already become intramural. Now, many of those agencies are becoming, they would support, but DST would support only the DST institutes, DBT would support only DBT institutes. So in the process, how are we? Because we develop a course and we take not many students. Are they industry ready? Are they capable of taking the challenges to take up things, to become an entrepreneur, to make a startup, or maybe that they are acceptable to the industries? So don't you think that this is the major issue, apart from the theoretical part we have discussed? Don't you think that this is something which should be debated and there will be concrete suggestions? How do you make them industry ready? How can we make them to go for entrepreneurial ventures or to make startups? Because that is the only future for the youngsters sitting in front of them.
I think it's a very, very valid question that Dr. G.M. Nair has raised. Are there any suggestions from the panel in this direction, or we can go to the audience and we can contribute in between as well? So, questions from the audience? Yes, please.
You have mentioned about the blood-brain barrier. The metastasis, the cancer cells are able to cross the blood-brain barrier and infect the brain. Whether we would be able to target those type of cells by specific modification of genetically modified viruses, wherein they will be able to deliver either the drugs or will be able to take care of such cells which are crossing the blood-brain barrier. And what is the mechanism by which these cells are becoming capable of crossing the barrier and reaching the brain?
So there are several ways of disrupting the blood-brain barrier or crossing is. One of them is a direct method. You can stereotactically implant viruses, attenuated viruses or chemotherapeutic agents directly into it. Then the indirect methods are, if you give mannitol, it kind of decreases the blood-brain barrier. So simultaneous giving of hyperthermic mannitol actually decreases the blood-brain barrier where chemotherapeutic agents start acting. Then there is a very interesting way of crossing the blood-brain barrier. If you encompass any medicine in lipiodol, which is lipid-based membrane, it crosses the blood-brain barrier. So the lipid membrane actually crosses the blood-brain barrier, whereas if it is not encased in lipid and it's a simple protein, it will not cross the blood-brain barrier. So that is a very commonly applied strategy to cross the blood-brain barrier and take it there. And the fourth important way is that a lumbar puncture is done. So a lumbar puncture in the subarachnoid space, the drugs like methotrexate are directly given to kind of bypass the blood-brain barrier. So these are some ways in which the blood-brain barrier is actually breached to give the chemotherapeutic agents in metastasis or in brain cancers.
Yeah, so there's
some good questions from the students.
yeah.
Hello. I would like to know what's the role of gut microbiome in human health. I heard that there are lots of impact of mimicking the gut microbiome of healthy people is a remedy for many of the diseases.
Absolutely. This is a very, very organized area of research. Actually, the maximum, we have lots of commensal organisms which are actually indigenous to the human body. I mean, every single centimeter square of our skin or any other part of the body has huge amounts of microorganisms, but they are commensals. And this is the common policy is that by changing the gut microbiota, you can actually cure a lot of diseases. Like nowadays, during my time when I was a general surgery resident, we would also often do a hyper-selective vagotomy for gastric ulcers. And then they realized that the ulcer itself, the biopsy, when they biopsied it, to their surprise that they found that there was a microorganism which was responsible, which was actually amenable to treatment by metronidazole. So when they gave metronidazole, the ulcers disappeared. And they found that, metronidazole alters the gut microbiota and actually causes these things. In many kidney diseases also, just by giving neomycin, it actually helps better in absorption of the relevant dietary things which will actually suppress the increase of urea and creatinine. So it's a huge potential, and I think this is a very, very upcoming area of science. Not only microbiome, the other very important thing is the gut has the largest number of nerve cell plexi. And now they are discovering there are more neurons and nerve cells in the entire gastrointestinal system than there are in the brain. And so that is another area of where there's a huge potential for research. So it's a lot of relevance there.
Antimicrobial resistance also is a big concern
Absolutely.
these days.
So,
So do you
yeah,
say
so
that?
so for example, antibiotic resistance is a huge concern, and this is exactly what happens that the microbe changes its antigenic character as soon as the antibiotic has been given for a long time, or it is stopped suddenly. So it is not given for the total duration of total duration. So somebody gives one, then stops it, then gives another one. The person becomes resistant to that particular antibiotic. That's why they say there is an antibiotic policy which has to be rigidly adhered to, which means if you are giving one antibiotic, then it has to be given in sufficient dose and for sufficient time so that it takes its effect and the organism does not develop resistance to it.
And I mildly object. I mean, no question that microbiome has a major impact on body functions. But I think there are also a lot of studies that exaggerate. I mean, the experimental model systems where you use abiotic mice, for example, which do not have any microbiota in the gut. They, of course, have also a completely different mucosal immune system. Using such models to explore the impact of the microbiome. And if you listen to some people, liver steatosis or fatty liver, it's completely due to gut microbiota. There might be an impact, yeah, for sure, but it's, I guess, not solely dependent on that. So I think one has to balance a little bit the views. Clearly an important, very important point, but one should be cautious, yeah, in over-interpreting some of the results. That's my very personal view, sorry.
I would just like to add, just out of general interest, I'm not an expert in the area, but I understand by weight, the microbiome on your body is about 1/60th. But if you go by the number of cells, it is 3 times the number of cells of the human body. So actually, we must wonder whether the microbiome is sitting in our body, or are we sitting around a colony of gut bacteria, microbes. So what I understand is that I do not know the exact balance between how the human life process is affected by this. But the study is very important, and our dietary habits, what I can recall, when I came here, I remembered the Professor Babu Nair, who was a former student of this university, agriculture university, who is a food biotechnologist, and who constantly reminds us, eat lot of taira padanganji, so that your gut microbiome is kind of healthy due to lot of stuff, junk food and medicines that we are eating. I just thought of sharing that information.
We can take up a couple of questions more. Right.
Good evening, sirs. I would take you to Kerala scenario where on biotechnology. I represent the government, the Kerala Biotechnology Commission, which focus on the biotechnology development activities of the state of Kerala. So the specific biotechnology development activities, all they rest upon the biotechnology policy, which was formulated way back in 2003, 2 decades before. And we have now started, initiated processes to update or revise the biotechnology policy, maybe a short 5-term policy for to see better development of biotechnology in the state of Kerala. So I would ask the panel if you could provide some recommendations, some key policy interventions that we can incorporate into the Kerala biotechnology policy, the 5-year policy, as an immediate priority, considering the Kerala's natural resources, the available manpower, the best brains that we have, but not industry ready. So,
maybe Dr. G.M. Nair, please, you can. The difficult question to answer.
I was also a part of Biotechnology Commission. So, what is ailing in the sector is that in Kerala, as she has rightly said, that we have a policy. Okay, people practice biotechnology. We have courses. We have human resource development, but then they are not skilled enough to go in. So one thing is that biotechnology will promise or progress only when you have industrial intervention. As a classical example, if you have 1 minute, I would just explain. I'm talking about a program called as Partnering Academic and Industrial Research, which we have promoted in KCSD. This is where an industry invests in research. A student is being supported through fellowship to do a research program in one of the major institutes, NIIST. And what happened was that we have several programs following that, but then the success story, the first one is that the student was given a fellowship by the industry. She worked on a problem which is relevant to the industry, and she got a PhD and she was absorbed back to the industry. How many programs are there which can follow this kind of thing in this? So we are into that where industry has to back the research programs in the state of Kerala. If the students have to find out a provision, a better placement, or maybe that they can practice it lavishly because there is no support that is coming even otherwise for the industry. So maybe that human resources development is one of the major programs which is envisaged that we cannot neglect that because biotechnology industry, industry ready human resource will have to be developed, and for that your curriculum has to be revised, has to be changed. Industries have not to come only for support of research, but also that they should be there in every sphere of activity when a program is completed. We are not ready. Even in the country also we don't have many examples, and I'm sure that over there, I'm sure in other places, in the outside the country, we have programs like that. So success would come in only when you have a purposeful research in biotechnology to be carried out. You need to have human resource be developed, who could take up an independent research program, a translational research for him or herself, so that it can be put into a startup culture. Dr. Sharika, I think that is one of the major points that to be revised in the policy. So if that is done, I'm sure that that would be better. But then we need to have that. Not only, that you have a lot of industry, BSc or MSc biotechnology programs to be started. Are we making them the right biotechnologies for the benefit of the society? That's what is to be done.
Yeah. I think the problem again maps to development of human resources or upskilling the human resources to be industry ready or to be ahead of industry. Probably that could be what academics should target to. They should look at the tomorrow's industry to be industry ready for tomorrow. I think this problem has been across the country. More than 50 biotechnology programs got closed in Andhra Pradesh and Telangana. Lack of students and outdated curriculum. So
also, sir, you need to work on an industry relevant program. You don't do something and say the industry to take it up. They are very choosy that way. They will never accept it. So find out, there has to be an interface between the industry and, I'm sure there are many people, Dr. Biju, Dharmapalan, I'm sure that he is into it, that kind of thing. Probably you can also answer how we can.
I think you should take initiatives in...
we have been taking it, definitely we are.
So we can have, please.
So just one, hello. Just want to say that GM Nair sir has mentioned regarding the syllabus. As a teacher, I have been working in this field. One thing that I have noticed is whatever syllabus that have been provided in biotechnology courses across the country in most of the universities are just a modified biology course. There is nothing like biotechnology. Technology part we are purposefully leaving for the sake of students. So this has inadvertently affected the job prospects. So this is one issue. The outdated syllabus comes because of this. And another issue is in biotech sector, as sir has mentioned, there should always be an industry partnership. But compared to IT sector, when students go for an industry, they get the salary very minimum. Even in Biocon, by MSc Biotech students get the salary in 20,000 or less than that. Whereas in IT sector, they get in lakhs. So this disparity often keeps away the students from going into the biotechnology field. So this is a serious issue. The industry should give good salary if good students are to be recruited in the industry. Thank you.
Few more questions. I think the students need to be asking more questions. Yeah, there.
Hello. Yeah, respected panelists and eminent speakers. Since we did touch the topic of lifestyle disorders, I just wanted to ask, since this talking about the declining fertility, and since we are approaching a state of infertility that has been seen in most of the women and men across the globe, I wanted to ask, how far are we in diagnosing such diseases related to fertility?
Any experts?
Wouldn't call myself an expert, but I mean, I think some of the reports, particularly male sperm count is declining. I mean, if you carefully look into these studies, and there's been also a lot of discussion about that, I mean, they are not as solid as they should be. On the other hand, there are also clear environmental factors. I mean, a lot of chemicals have hormone-like function. Yeah, so if they are, for example, from these PET bottles, there are some residual chemicals released which have an estrogen-like function. And have been a lot of speculations that this also interferes with fertility. So these environmental factors, one can surely do something about it. Be there more strict what's released. Other studies, again, I think one should be careful really looking into that. It's also the tendency that females get pregnant later. I mean, this will anyway go along with a certain decline of eggs. So I think this has to be really carefully evaluated. There have been a lot of reports. I'm not so sure. There might be rather, in my view, environmental factors which are also straightforward to fix, straightforward in principle, but yeah, bring industry and all of us to a situation to take more care of these chemicals that are released every day. That might be a big challenge.
Any other thoughts here? So in fact, the large number of in vitro cases increasing in the country is one of the reasons. And so maybe microplastics could be one sole reason. I mean, are there further questions from the audience?
Sir?
Yeah, please, go ahead.
Sir, as we all know, biotechnology science is very fascinating, time consuming. Can we make it more affordable? Like from the point of view of a common man, if he want an organ transplant, he have to depend on the stem cell therapy or stem cell banking or the 3D organ printing, which in turn is very costly. So how can we make it more affordable? Is there any studies going on to make it more affordable for a common man?
Yeah.
I think not at present, because I say most of those things that is being done are all expensive things. It won't be to the reach of the common man. See, this has to be done in a large way. If you need to reduce the cost, input-intensive programs are being practiced. So means that the product also are going to be expensive. See, a classical example is that a drug, you know that 13 years it takes for a drug to be identified, developed, clinically tried and put into them. And maybe at the 13th year, you would realize that the drug has some side effects or some problems. You withdraw the drug. Who is going to bear the expenses of that 13 years of failure? So in that case, they book it to the customers. So those who are taking the next drug will be priced twice, incorporating the failure that they had. So likewise, if in this case also, I'm sure that this is going to be a great thing, provided, and if it has to reach the society, I'm sure Dr. Sanjay also would vouch to it. If it is practiced by a multinational company and done in a bigger way for the benefit of people, probably the price would come down. Do you agree to that?
No, actually the price is initially high for many reasons. The first reason is that it takes almost 15 years for a pharmaceutical product to develop. And it takes about 15 to 20 years because, like I told you before, there are two ways. One is you keep on trying something until it succeeds. Or the second important way is that if there is some background knowledge about a particular product, and then you go on developing it, look at it from a very, very basic science point of view. then you might get some hints into how to develop it. So that's the first step. Then as you go on developing it, there are several ways, even if you get some hint that it is successful, then it requires a lot of trials, lot of, for animal trials, it requires human trials, where even a small adverse event has to be recorded and the patient compensated. So by the time they finish the entire trial, it takes so much of time and expense that they would want to recover the money in some way, and then as it becomes more and more popular, the price of the drug goes down. So that is the way, that is why, you know, and then there is a intellectual property right on that drug. So for 10 years, they can actually use that intellectual property law to get some profit out of all the expenses they have made over several years. But then if somebody is successful in making a drug, then the company which buys it, it goes into billions of dollars, not millions, but billions. One drug will, you know, just buy the entire company. That's the way. The second thing is, ma'am, coming to your question, I'll just tell you, there are 5 ways in which you can enhance your, improve your startup, scenario. The first is, start with the end user experience. Like for example, ISRO and everybody, they don't say, okay, scientists, you make this product and then we'll see where it works out. They give a specific problem, and based on the problem, there are several people who are working on that specific problem, and when one succeeds, then the person is given that contract. So the first is an end user experience before or at the start of the project. That's the first thing. The second very important thing is transporting to scenarios where things actually are happening. Like for example, if you are asked to make a product, then actually coming to a hospital and seeing the scenario in real time and for several days and months suddenly gives you ideas which are completely different from the way a doctor will function. That's the second point. So that is the second thing, transporting to scenarios which are intrinsically useful for understanding the product. The third very important thing is developing ancillary skills or inheriting ancillary skills from other organizations, which means if you're not computer savvy and you are doing some product, then you have to hire people who can actually help you in that. Or if you're doing a large data study, then you must hire a statistician to help you work on that large data epidemiology. And design it, for example, if you're designing a product, then you have to hire a designer to work with you to get it done. So these are developing ancillary skills, very important. What happens is, even if you make a product which is intensely successful, its design is so bad that nobody accepts it. So even if you want to sell something, then you have to, say if you go to a party, you'll dress up well, right? So that's the way it is. You know, you have to, the design component is missing from our products, which needs to be focused upon. Then the fourth is some kind of rational funding. Most of the funding is totally irrational. They just say, okay, how much money do you want? Okay, this is the money you want, get this much, 30 lakhs, 40 lakhs. What are you going to do? What are the and variables? I think Sir will be able to also talk about it. But what is that rational funding that you need to get at that particular point of time? That's a very important aspect of it. Now, supposing I don't have a background and you give me 5 lakhs rupees. It's the wrong thing to do, because neither do I have background, nor do I have mentorship, and you are just wasting your money and decreasing your translation rate on something which will never be done. So therefore, this is a very important point. A rational funding is important. And the 5 important point, which I think most people don't understand, is you cannot expect a pure scientist who has spent all his life doing science to actually sell his product. So therefore, you need experts who are in sales and who are in different intellectual property and I think to actually do that work. So they are your very close collaborators. Here what happens is, you think your scientists are your collaborators, but the person who's actually selling the product is some useless person who can be given something and that's the end of it. No, he has to be intimately involved in your project. So once you actually have these 5 things in place, then a lot of things get done.
So I think we have seen a different role of Dr. Sanjay here. So he has almost summarized the entrepreneurial journey in 5 points here. So while casually he was walking, he said, I want to do new budding and grafting and all. I said, it's retirement plan. Now I found that he has a very clear track where he could really look at how an entrepreneur can be budded out.
And if you ask me one more point, coming to this one, you must always have multiple hobbies. So that, for longevity of your passion in a professional field, you must have maybe read or music or some aspect or gardening or something which actually keeps the stress away.
One of the key points in the beginning is how to introduce this immersion experience in our curriculum. So taking the students out to the industry, out to the clinics, understand the problem, empathize with the people. Empathy, which is the core of the problem. Once you do that, then you probably can innovate on the ideas, form a team, bring funds, and all those things he has explained. So I think we will wrap up this discussion here. Thank you very much for all who participated, asked questions. I know there are a lot of questions, even I had a lot of questions here. But as a last session, I will probably step down from being a moderator. I would call up one of the young enthusiastic rapporteurs all around here. So one of you would come up and probably quickly summarize the key points today. So there have been a good rapporting out here. So who want to come up, please? Good name? Yeah, please walk up to the stage and you can quickly brief about the... Yeah, good name? Aparna. Okay, please, Aparna. Yeah.
So, hello everyone, respected panelists. My job is to brief the panel discussion. So the question raised by the question we started was, what is the next generation of researchers to be, and what are the economical assessments and advancement in biotechnology that has impact in your life? So the first one to start with Dr. Sagar Pandit. He's a plant science scientist from agriculture biotechnology background. So according to him, biotechnology is a field which has pushed agriculture rather than any other fields. So we have BT cotton, BT brinjal, but it masks the other areas of biotechnology in agriculture. We are not ready to accept the GM crops because of the ethical and political issues, and the research is also time consuming. And next was...
yeah, probably you can summarize each question with the essence and okay, yeah.
Just
the quick points, yeah.
Professor Dr. Thomas Braun, we are not ready to take risk. COVID pandemic has fast development of immune technology, lack of information, modification, mutagenesis, CRISPR is a challenge, opportunity to young scientists to come up with crazy ideas, create innovations and accelerate further in the future. Professor Dr. Sanjay Behari, in medicine, nutrition and agriculture, stem cell development, diagnostic chip can be used for transplantation, AI, artificial intelligence, and nanochips. Professor G.M. Nair, research should reach society. We should be more prepared, and prevention and management is important. Abiotic and biotic stress of plants, reduce compartmentalization, interdisciplinary, contribute something to society. Professor Dr. Achut Shankar sir, biotechnology is a maker of things, rather it is different from biology, biochemistry, biophysics, lab, meat and BT cotton, issues in the fields, environmental, ethical, political, and synthetic biology is a field. Professor Dr. Soni KB, transgenes is a debatable topic, GMO is still growing, RNA technology has less issues, and also nanobiotechnology. Bioinformaticians has come up with Human Genome Project and CRISPR. Limitations, public trust, environmental, lethal, legal, and socio-economic aspects. about gene editing, Dr. Soni ma'am has said it has been mostly used. Technology is clean, but monitoring is required. Then immunospecificity and immunosensitivity, Sanjay Behari sir. Immunotherapy, immunomodulation, suppress autoimmunity for organ transplanting, passing immunobarriers to destroy tumor, pharmacogenomics, exploring blast by Achut sir. Dr. Thomas Braun sir, static targets are easy, but moving targets are difficult. Inflammation is a double-sided sword, precision medicine. Sagar Pandit sir, climate change versus anthropogenic changes have an increased effect. Professor GM Nair sir, quality of education, industrially ready students. Question.
Yeah, I think that's fine. Yeah, yeah. Thank you, Aparna.
Thank you
very much. So, any quick summary points which we want to add from the panel? Yeah, yes, sir.
The only thing I just want to, say in conclusion is that Trivandrum is so lucky to have so many institutions, national, state institutions in the vicinity, 15 institutions. The biotechnology programs conducted in different places should have a kind of interaction with all these institutes. Industries are there. There are many industries who are willing to cooperate. But the only thing is that how do you make it open? How do you make an interface between all these? So Institute Sri Chitra, there's a classical example of where technology is being transferred lavishly. NIST is a place where so much of, they should know what is that technology transfer. All those, unless you have an interaction process, they should be brought into the institution to take classes, not just an organized lecture. And then if that kind of involvement is there, I'm sure that things would improve. So this is one of the major things that should practice, that make license, not only with the industry, but with all the institutions practicing biotechnology in the vicinity. There are so many institutions, but I'm sure Trivandrum is very lucky that we have so many institutions. So maybe that would be a last take-home message they should think. Interact with people. Standalone biotechnology has no value. You need to interact, it should be really interdisciplinary, and then what lacks in you can be complemented with any other person if you make an interaction. Just a take-home message.
Actually, what we don't realize is that in everyday things that we do, biotechnology is already prevalent. It's just that it's improving by the day, and newer things are happening. So cohesive interdisciplinary strategy and understanding that high quality products require a high degree of validation is the need of the hour. What we often neglect is the degree of validation that is required and the degree of testing that is required in order to make that product successful. We have to give adequate focus on that also.
Thank you.
Most of the discussion has been on whether the biotechnology is sustainable, whether it is affordable. It's not just about the biotechnology and the products, but even the biotechnology education. You mentioned that the syllabi are not updated. Most of the syllabi are biology syllabi. I'm also involved in such discussions back in Maharashtra where I work, and also at let's say a bit in the national level. The limitation here we have identified is, number one, if you have an updated syllabus, do you really have faculty to teach it who are capable of teaching such a syllabus? Number two, if you really have that updated syllabus, do you have equipped facility lab in your place which is teaching this biotechnology? And more important, biotechnology means something different to everybody. If there is a biotechnology subject which is being taught in the institute which has plant biology dominant, they think everything is plant biotechnology, is all biotechnology. somewhere where there is a biomedical sciences dominant institute, there it is, biomedical sciences, biotechnology dominant. So I think it's a time to ask ourselves a very hard question, kind of introspect, whether we really want to carry on with this biotechnology. Is it real or it's just a bubble? I'm not saying it is. I'm saying we should ask ourselves. And, maybe think about decentralization. Leave the biotechnology of neurobiologists with neurobiology biotechnology, the one with plants to them, and so on. That might also be a very useful approach at certain times. But I think just being emotional about carrying over with the biotechnology may not help certain places. And that is the thing that the policymaker should very realistically introspect, is what I would like to sum up.
Thanks. Thank you. Yeah, so I think very valid question on the curriculum revisions, but we really need to look at the interdisciplinary nature, bring in courses of value. So I'm pretty sure no biotechnology programs have an AI or ML course right now. So do we need to bring in such courses to bring the interdisciplinary flavor? Of course, curriculums have to be revised with the industry perspectives in mind. Why don't we call industries to ask what do they want? So we make our students industry ready, we make ourselves industry ready. So we need to upgrade ourselves to teach to the next generation. We need to look at new courses, interdisciplinary nature, so that, we don't really talk about biotechnology as a bubble, but we really need to upgrade ourselves so that this is the future. So
curriculum need not be teacher-centric.
Correct.
If the teacher has no expertise, he has to learn it, because that's why he is the teacher. So it should be student-centric, industry-centric curriculum that is to be developed, isn't it?
Right, so thank you very much once again. I would like to thank all the panel members today who have been contributing to the session, adding valuable points. I would strongly urge the KAU team to sort of relook into some of these topics. Let us come up with a proper document. We will be more than happy to contribute more into these thoughts. Make it as a white document, and we could submit to the policymakers, to the Kerala government, to the central government, so that we can take up some of these agendas ahead. Thank you very much once again. Thank you to all the audiences.
Thank you. Thank you also for effectively moderating the session. Thank you. It was wonderful.
We would like to extend our thanks and gratitude to our panelists for being here. The topic was covered from all disciplines, and it indeed was quite informative and interactive. We thank you all.
Now, without further ado, as a token of appreciation, we would like to present to our panelists with a memento. For the memento distribution, I would like to invite Dr. Roy Stephen, Dean of Faculty, College of Agriculture, Vellani. Sir, please come to the stage. I request Dr. Rajiv K. Sugumaran to receive the token of appreciation. Thank you, sir.
Next, I invite Professor Dr. Soni KB to receive the memento. Thank you, ma'am. Up next, I would like to invite Dr. Achut Shankar to receive the memento. Thank you, sir.
Next, Professor Dr. G.M. Nair to receive the token of appreciation. Thank you, sir.
And last but not the least, I'd like to invite our moderator of the panel discussion, Professor Dr. Renu John, to accept the memento. Thank you, sir.
I would like to invite Mr. Saga Pandit to receive the token of appreciation.
Thank you all. Today's panel discussion was a resounding success. We extend our heartfelt gratitude to all who attended and contributed to this enriching discussion.
I'm sure this event has introduced each and every one of you to new disciplines and arenas of ever-advancing field of biotechnology and its future possibilities at 2047. We saw our panelists' perspectives on different branches of biotechnology and related fields, including agricultural biotechnology, development of genetically modified organisms, bioenergy, and creating transgenic energy resources, biomedicine, tumor studies, and new technologies like space medicine and also genomic studies.
Throughout this conclave, in the coming days, we'll have a chance for a collaborative learning of scientific exploration. I hope our first day at BioZion has set the tone of enthusiasm for exploring and exchanging of cutting-edge ideas and research.
Once again, we thank all our honorable guests, panelists, moderator, teachers, and students who actively participated in making today's event a resounding success. I'm Shweta Gautam.
This is Elizabeth Jeevan Philip.
Yeah, we both are students of BTech Biotechnology, College of Agriculture, Vellani.
Thank you.
Thank you. The cultural events will begin shortly.